Welcome to the show, Lindsay. Great to have you.
Yeah, I'm so excited to be here. It's been a long time coming.
100%. I would love to know more about you. Do you want to start with a quick introduction?
Yeah. So, quick intro. Growing up, I was a military brat. My dad was in the Air Force. So, we kind of spent the first 16 years of my life moving around every 2 to 3 years. We finally kind of landed in New England when I was in high school. And it was around the same time that I started reading this book of poetry by a kid, Maddie Stepanek. And he had like this really awful disease, a disease where he didn't have energy, basically, like his mitochondria weren't working. So he couldn't make energy, not just to kind of like go for a run or go play, but just for life. Life needs energy. And I got really fascinated with molecular biology after reading that book of poetry. And I was going into like high school chemistry. I was so nervous about it because I was not the best at math. I wasn't doing great in calculus and algebra classes. So I was like, oh man, there's no way I'm going to survive chemistry and physics where there's so much math. But I had a fantastic teacher who just really kind of broke it down. And that really is where my love for chemistry kind of sparked and biochemistry. And I wanted to go get my degree in college. And I specifically wanted to do biochemistry, molecular biology. I didn't want a biology degree with a couple extra classes in that space. And I didn't want a microbiology degree with a couple extra classes in that space. So I found a program that offered exactly biochemistry and molecular biology as the actual degree and did some undergraduate research and didn't have a great time. I did not have a great time in undergrad doing undergrad research. And I must have been like 20, 21 years old. So not even a quarter life crisis, like a fifth life crisis, a quantile life crisis. Because I thought I wanted to be a scientist ever since high school. At that point, it had been a couple of years, thought I wanted to do science. I had a bad undergrad experience. So I ran away and I thought teaching might be what I wanted to do. So I went abroad on a Fulbright scholarship and taught English as a second language in South Korea. And it was awesome. I loved it. I loved teaching. My high school kids were hilarious and awesome, but I really missed science. So I came back. I moved to Boston. I found a job, got really lucky finding a job. This was in 2011, and I liked that for a couple months. It was a genomics job. And after a couple months, I realized I didn't really like genomics. So another like quick crisis of, oh my God, what am I going to do when I grow up? Found a proteomics laboratory and in like total transparency, this was 2012. And the woman who had later become my mentor looked at my resume, looked at my CV and told me it was awful. She was totally honest. And she said, if I just saw this CV, I would not have hired you, but you came recommended by someone that I trust. So I'm going to take a chance on you. She took a chance on me, fell in love with proteomics. I realized I just wasn't doing science with the right people before. I wasn't doing science where I was intrinsically just interested. It had nothing to do with how like smart you are or inherently like talented or gifted. It was what drove you, like finding that driving interest. And it took me a long time to figure that out. Once I found it, I was like, I'm going to grad school. I'm getting a PhD. I'm going to teach myself how to program. I'm going to like learn how to code. It's going to be great. And it's been awesome since then. So just anybody out there who's maybe like struggling with that quantile quarter life crisis, wondering what you're going to do when you grow up, that's totally fine, totally normal.
Yeah, it takes time to find your sweet spot, what works for you. So you have to experiment a lot, right? You have to try
absolutely
different things. So what is it about proteomics that you like so much compared to genomics, which is way ahead? And if you talk about like you can do the whole genome sequencing less than $1,000 now.
Oh yeah, and I was working at the Broad Institute, so the whole human genome sequencing project was like so big. They were so heavily involved with that, and I just honestly didn't find my drive in genomics. A lot of people do find their drive in genomics. For me, there wasn't quite enough chemistry. With genomics, a lot of those experiments are kind of like you buy a kit from Kyogen or something and you just mix the kit together. I wanted something that was a little bit more process intensive, like a little bit more chemistry. And that's exactly what proteins give you. DNA has 4 base bases that pair
Right.
together. Proteins have 20 plus amino acids. Those amino acids can be modified. They can be rearranged in structural space, 3D space. And that was just kind of, I thought that was so cool. I really loved kind of the protein chemistry aspect of it. And I really liked the fact that, kind of going back to that first inspiration, that kid, Maddie Stepanek, it was proteins, right? It was proteins that were causing disease. It wasn't necessarily something wrong with his DNA. It was these proteins had issues. Could you find protein-based therapies, protein targets for therapies, really is what drove me.
Love it. Love it. I do think what happened to genomics last 2 decades will happen to proteomics. Like there's a lot of work to be done in the proteomics space, right?
Oh my gosh, I totally agree. I totally see where genomics was 10 or 15 years ago. I think that's kind of where proteomics, now we have some techniques where we can really start scaling up, and it's just starting to take off now. So it's an exciting time to be in proteomics, I think.
100%. Couldn't agree more. Do you want to talk about your entrepreneurial journey? How did you end up as the CTO of Talus Bio?
So I decided I was going to go to grad school, and I decided, proteomics was where things were going to start happening, and specifically going to like larger and larger scale proteomics. That's where biomed research was going to go that direction. So I decided to be joint advised. So one of my professors was a computer science machine learning expert, and this was a deliberate choice by me because I wanted to learn programming. I had never programmed like anything. I had tried to do a Python hello world script before grad school, and I couldn't even get that to work. So like we were really starting from nothing going into grad school. And this guy, Bill Noble, his lab does machine learning. And kind of the same thing when I decided to move to South Korea. I wanted to learn the Korean language. What better way than to literally just move to the middle of nowhere in Korea, and then you're forced to learn it. Exact same thing with this computer science and machine learning lab, right? Like if you want to learn a language, Python, coding, just total immersion. So it was a steep learning curve joining that lab, but so worth it. So I was joint advised by him, and I was joint advised by mass spectrometry proteomicist, Mike McCoss. And 2014, I started grad school. 1 year later, a guy, Alex Federation, also joined that mass spectrometry proteomics lab. And he was just like the kind of coworker, the kind of colleague where you can be in the lab doing experiments like all day, troubleshooting experiments failing. Like you can be in the lab for 12, 16, 18 hours. Just slogging through the science. But then the kind of person you would still want to go get a beer with afterwards, even though you had such a long, rough day, like you still just want to go out and talk science and hang out. So met him, we started working on a collaboration together, and that collaboration is what built Talus's platform technology.
Love it. So you found your co-founder.
Yep.
What next? Like what challenges, what type of barriers you had to cross to reach at the current stage of the company?
Yeah, so we were doing work. I was at a university. I was at University of Washington. He was kind of joint appointed, kind of between University of Washington and at the time new institute called the Altius Institute. So we were kind of working in these like academic nonprofit spaces where the technology was developed. So obviously when you're starting a company, kind of having intellectual property, having IP is very important. Right. So the Altius Institute was interested in patenting that technology. So obviously if you're trying to get into a patent space as an academic, it's very frustrating because you're really not supposed to talk about the technology at conferences, and you're not supposed to like publish on it or anything. Right. So it can be very, very frustrating when you're a grad student or a postdoc and you're sitting on something you think is really cool. You can't really talk about it because you got to get that patent submitted first. So that process kind of slowed down disseminating the technology when we were working on it. The patent got kind of written up and worked out with the Altius Institute. So we were able to publish the paper last year, 2020. And it kind of flew under the radar. When we presented the technology, we didn't really focus on the applications of the technology. We just focused on like, here's a way you can study chromatin biology. So it kind of was quiet for a little while. And while it was staying a little bit quiet, my co-founder, Alex Federation, took advantage of, there's various sites across the US that are embedded in academic institutes like universities, colleges, and provide entrepreneurial education, right? So National Science Foundation has the I-Corps program. They kind of offer like workshops and seminars and kind of boot camps for exactly this situation. You have some academic project that you want to spin out into a company. How do you do that? You need business plans. Is there a product market fit? Does anybody even care about what you're doing? Coming up with a first pitch deck. How do you find funding? All of those questions that you have when you're just like, I have a cool idea. I think it'd be a cool company. How do I connect those two steps from cool idea to cool company? Those programs are very, very helpful. And I think they're all over the place. Berkeley has like iGEM, I think.
iGEM is also global now.
Yeah. So I think if you're an academic or nonprofit, you're like a trainee, a grad student, a postdoc, even early career, like professor, you have an idea that you want to commercialize into like kind of a company, take advantage of those programs, take advantage of your school of business at your university, because they almost certainly have people who would be good co-founders, not a technical co-founder, but like a business savvy co-founder. So I think those are kind of good first steps if you're kind of exploring whether you want to spin out from an academic institute.
Love it. So you need a founder. You found the founder. You need to protect your IP. It's protected. Then you can talk about it. Then you have to see if there's commercial viability, right? So for that, you need help. As a scientist, you're not trained like MBAs. So you have to figure that out, right? So I couldn't agree more. Like incubators within universities, business programs, they're helping create more scientist entrepreneur. And I think we will see more and more scientist entrepreneur. Historically, we have seen entrepreneurs more businessy, right? They're trained in more finance and know all the game. But now we are seeing entrepreneurs who are tinkerers, right? They are inventors. So it's really exciting times.
Oh, yeah, I completely agree. And I think a lot of scientists don't think of themselves as an inventor because they're thinking like, oh, well, I didn't invent anything. I just like wrote a protocol. It's like, that's an invention. You invented something. If you built a protocol that never existed before for a sample that never existed before, anything like that is inventing. That's an invention.
Right. And especially now, it's clear that bio revolution is on the horizon, right? We need more bio entrepreneurs than ever before. So
I totally agree with that. And that kind of is like the next stage for my company. So the patent has been like kind of worked out. So we were able to publish the paper. 2020, my co-founder kind of does one of these boot camp kind of workshop programs to do some interviews, reaching out to biotechs, pharmaceutical companies, kind of asking like, I have this kind of idea. Is this something anybody would be interested in? Establishing that there's at least some product market fit there.
Right.
So then the next step was he saw that there was some interest there. He saw that it was a cool idea. He kind of did some of his first steps. So his next step was to find co-founders. So I was one of a handful of people he reached back out to. And obviously, since I had such a great time working with him before when we were inventing the platform technology, I was like, oh my God, hands down, yes. Those kind of coworkers, those kinds of collaborators are so rare to find. And I was so excited to embrace the opportunity to work with him again.
100%, because a startup journey is already so hard. So last thing you want to do is work with people you don't like, right? So.
Oh my gosh, absolutely. And it's kind of, having that little bit of like that trial period where we had those really long days in lab, where we had opportunities to end collaborations and just walk away. But we just kept going back for more and more ideas, more and more like, we have no extra time, but let's add another side project just because it would be cool. Finding someone that you fit with like that, I think is really important for co-founders.
Right.
And it establishes that level of trust too, because you've really got to trust them a lot.
100%. I do want to talk about Talus Bio, go deep in your technology, what is unique, how it is innovative. But before that, I want to get some input from you. Like, what advice would you give to aspiring bio entrepreneurs? Still a bench scientist at university, a postdoc student, PhD student, what should be their approach? Like, how can they do all of that you managed to do?
I think if we were maybe more intentional about Talus as a company from the start, probably what we would or should have done is gone to that tech transfer office. So we went, Altius was writing up the patent, so that was a good first step to make sure that the IP was protected. What we probably should have done then is started looking specifically into those incubator style programs that you had mentioned. So at a tech transfer office, for example, at University of Washington, and seeing if we could get space in their, entrepreneur program in their incubator space. I think those opportunities are really nice. If you can find them at your institute, having their support is very helpful. So that probably would have been good first steps. I also wish I had known about some of the grant support that exists out there. So there's, when you're an academic, you're a bench scientist, a lot of times I think we know about the more academic grants like an R01, or if you're a postdoc, an F32 or a K99. And we are aware maybe of those mechanisms, but there's lots of other mechanisms specifically for businesses. So the small business SBIR grants, those would have been good to start thinking about earlier for us. I wish I had known there's also transition grants. So like the K99 grant mechanism is, okay, you're a postdoc, you want to be a professor. So the K99 will fund part of your postdoc, and then you'll take the rest of the money into your professorship. They have similar things for entrepreneurs where you're a postdoc or a grad student and it'll fund some of that. And then you take the rest to the company. So like a small business entrepreneur version of those grants, like I just didn't know about these opportunities that existed. So we didn't even know what to take advantage of. We just kind of fumbled through and then found out about them afterwards. So yeah, I would say reach out. I can almost guarantee your university, probably even your department, has someone who did this before. Probably they don't talk about that person quite as much as they should have because they're training you to be an academic, not to be an entrepreneur. Find out who those people are and reach out to them and ask them specifically for your department, for your lab, for your university, what resources are there for you to spin out?
I agree with you. There are so many resources within university because if you start a lab, it's not inexpensive, it's very expensive, right? But university has all the resources you would need to start a company. And tech transfer office is also very complementary in nature. Like they only focus on the commercial viability. You only focus on science. So joining forces with them moves the needle quite a bit.
Totally agree. Totally agree.
Yeah. Cool. I want to learn more about Talus Bio, a fellow Y Combinator company. So I want to understand more about it. What is the mission, vision of the company, and also want to talk about the science more.
Yeah. So Talus Bio is a platform drug discovery company specifically for what have been previously called undruggable proteins. So pharmaceuticals are mainly interested in using our kind of technology to improve the drug development pipelines for proteins called transcription factors. And these proteins, transcription factors, when you're born, you have a genome, you have a set of DNA, and as you grow, as you develop, that same DNA is used to create your skin cells, your brain cells, your muscle cells, your nerve cells, obviously all very different cells, but coming from the same DNA. So transcription factors are the proteins that read those different genes and will turn on or turn off genes in order to develop these different phenotypes. So that's great. That's kind of like what happens when everything's going well, right? You're differentiating from stem cells to T cells or muscle cells or skin cells. But what can go wrong is the wrong transcription factors turn on at the wrong time, and that can cause diseases like cancer. So Talus is working on using our technology that reads what transcription factors are binding to DNA and using that information to develop drugs for those transcription factors. We want to be able to switch off the genes that are causing cancer, maybe switch on the genes that would rejuvenate an immune system, and build a better way to make therapeutics for these proteins.
So you are saying when something happens, whether it's epigenetics, environmental, nature impact or nurture impact, transcription factors activates wrong genes or deactivates wrong genes. So what you are working on is making sure you fix that problem. Okay.
Exactly. So it's not like CRISPR. We're not going to edit the DNA itself. The DNA is going to stay the same, just like it's the same in all of your other cells in your body. We're going to keep the DNA the same. We're just going to figure out how to turn on and turn off those transcription factors.
How will you do that? It is just by working on their binding, where they bind, what are the binding sites, what are the receptors, or it is based on you change the structure so if they bind at different places?
Yeah. So we are going to do all of it. So the idea behind the platform is rather than focusing on a specific binding pocket. So a lot of other drug discovery, drug development approaches focus on, I have a structure for this protein and I know the protein structure, therefore I can just try to find binding pockets on that protein structure and fit a small molecule into that binding pocket. That works great for some proteins where you know the structure.
Right.
Transcription factors are not those proteins.
Okay.
Transcription factors, we do not know their structure. We know part of their structure. Most transcription factors are, of course, binding to DNA. So that part of the protein, we know what that looks like. We know what that DNA binding region of the protein looks like, but everything else we don't know.
Right.
And the DNA binding portion of the protein is not a good drug target.
Right.
So a lot of kind of traditional drug discovery approaches have deemed transcription factors undruggable.
Right.
They're not actually undruggable though. We've been calling them undruggable because we can't use our current approaches to drug them. These current approaches rely on knowing a protein structure, being able to crystallize the protein. And we can't do that with transcription factors. When we take a transcription factor, when we think about the transcription factor kind of in its native environment, it's in a nucleus crowded with DNA, other proteins, interacting with these other proteins and these multi-protein complexes with multiple kind of binding partners, right? That this protein is physically close to. So if you take that protein outside of that context and just put it in a test tube, it kind of falls apart. Like it doesn't have all of these external factors that help it keep its shape and therefore its function inside the nucleus. You take it out of the nucleus, it has lost its function, it's lost its structure. You can't do drug discovery on that anymore. So Talus kind of asked the very, simple or elegant question of, if you can't do this in a test tube, just do it in a live cell. So we're doing all of this drug discovery. We take a snapshot of all the proteins interacting with DNA before and after a drug treatment. So if a drug affects the binding of a transcription factor to DNA, it's inhibiting that transcription factor, it's degrading that transcription factor, that's what we measure. So now we can do these high throughput screens, we can test all these compounds and ask, does this compound specifically and sensitively knock a transcription factor off of chromatin? If it knocks a transcription factor off of chromatin, that could be a good lead for a disease in which that transcription factor is causative, like cancer.
I know it's a platform play, it's a platform approach, more agnostic, but are you focusing on one or two disease areas or therapeutic areas in the beginning? And like, how do you choose where to focus? Because it can go in thousands, if not millions of different directions.
Yeah, that's definitely one of kind of the hard parts about a platform company. And I think maybe there's a lot of bench scientists out there who have discovered, like invented protocols or methods or platforms like this. And it's one of the struggles, right?
Right,
How do you decide what you're going to do with it when you can do anything with it?
right,
So my co-founder has training, has experience in epigenetic drug development for cancer. He was trained in a lab in Boston under a guy, Jay Bradner, who's now at Novartis, quite prominently at Novartis. So Alex, my co-founder, had experience in this space. So it seemed like a very natural fit for the
right,
first thing we're going to go after. We're going to do cancer therapeutics against transcription factors in cancers that we know are caused by transcription factors.
right. And how much are you leveraging more in silico tools or in silico modeling, or how much of the work is done at the bench? Because I think there's a lot to be learned from the existing data than to create new data.
Yeah, I totally agree. And I think that is also a trend that we're seeing. I think trends that we're seeing in biotech startup companies and spin out companies, I think we're seeing trends in platform based
Right,
companies. And I think the other thing we're seeing is this same kind of non-committal approach to wet lab versus dry lab. I think a lot of biotech companies are appreciating that you are better if you're not relying on one versus the other. So we have a pretty equal split between our wet lab and our dry lab.
right.
And we are using some in silico approaches to do drug discovery. But again, a lot of those approaches also rely on having a structure for the protein. So we're kind of back at square one with some of those approaches because transcription factors don't have structure. So we can't use those structure-based drug designs.
Is there any database to find all possible transcription factors and all of their details about their properties and some information about the structure?
Yeah, there's not great resources for that. Probably the best resources that get at that kind of question are like the ENCODE project. There was
Right,
this really big consortium and they're doing kind of chromatin atlasing basically across a lot of different cell lines, different cell types. That gets close, but those approaches are pretty much all sequencing based.
right,
And we're really agnostic to the sequence itself. We're really interested in these molecular machines that are reading the DNA, that are editing the DNA, that are manipulating DNA. So they have complementary information in those databases, but really we're building the first one that is a protein centric, a transcription factor centric approach to this.
love it. Okay, so you have a solid technology, you have a solid business funded. And what is the business model? How do you plan to make money in the short term and the long term?
Yeah, so this kind of picks up the story where we left off in late summer of 2020. So now Talus has formed. I've come on as a co-founder. Alex and I are equal co-founders. He took a CEO position. I took a CTO position, and we have established that there's some interest here. The next step was fundraising. Can't really do a lot in biotech unless you have some money for lab space and lab equipment. So we started doing a pre-seed round of fundraising, and it was a lot of just cold emails, cold calls, using warm intros where we had connections. Alex got a couple of those warm intros, those connections through one, he did that program, that summer program, the I-Corps program. But two, he also, in the time between when we worked on this technology and Talus was born in 2020, he had been working at a startup in Seattle. So through that startup, he also kind of learned what things to be thinking about and just kind of like the vocabulary, the language again, right? Like the best way to learn any kind of language or technique is just total immersion. So he got the total immersion into some of these circles, these networks by working at that startup. So it was a lot of just cold emails, cold calls. We got a lot of no's. We learned a lot through those calls because we were told by investors point blank that, as a platform technology, you need a better business model. You can't just say we're going to do all the things with our technology. So that's when we arrived at this other kind of growing trend in biotech these days, which is the hybrid business model, right? So like
100%.
platform companies becoming very trendy as far as like in the biotech company startup company space. Platform companies are getting big. Having a blended wet and dry lab approach is pretty big. And the hybrid business model, these seem to be like
Right.
kind of the recipe for a lot of biotech startups these days.
So
So
when you say, sorry, I didn't mean to interrupt. So when you say hybrid, you mean CRO services plus your own drug candidates?
yeah. So kind of towing that line. We're not trying to become a CRO. We're not
Right.
trying to become a fee for service style company. But with a platform, you can do that fee for service work to start bringing in a little bit of revenue to help defray some of the costs. You're also being smart about who you take on as contracts. You can also start building relationships with bigger biotechs or bigger
Yeah.
pharma companies and hopefully prove to them through these fee for service contracts that your platform adds a lot of value to their drug discovery pipelines. If you can point out like your big pharma company already has, tons of scientists working on this, but we provide data or we provide information that you cannot otherwise get, and it saves you time or it saves you money. Having that kind of proof of concept style fee for service contract can be helpful in building that relationship, building trust and connections in those pharmaceutical companies that can be very strategic in your own drug discovery pipelines.
I love this approach because I think the fastest way to penetrate bigger companies is you start with a service, you gain experience, you build relationship, and then you productize based on your learning, right? Then you have
Exactly.
your own product in the pipelines. It's one of the greatest way to scale as a company, and
Yeah.
also de-risk along the way.
Yeah, I absolutely agree with that. And I think that is kind of for biotech companies, I think that's a good way to interpret what a lot of like the software companies and what we learn at Y Combinator when they're telling you get that first beta version out. Get
Right.
some beta testers, get some customers, get some users, and then iterate your product based on what they're telling you, the feedback they're telling you. So in biotech, for a platform-based company, using this hybrid business model gets you at that same kind of strategy, I think, where you're hearing from bigger pharmas, you're hearing from biotech what they need out of a platform. You build your platform or extend your platform or optimize your platform to give that information. And then you just keep iterating like that until you end up with a platform that is just an absolute powerhouse for what you want to do.
100%. This agile approach, working with your potential buyers, really help you create a product that people want and it will be usable by others too. So what will be the final product like at Talus Bio? Will it be more like a reagents material, or you are working on your own drug candidates? What will be the final product here?
Yeah, we're working on our own drug candidates. So
Okay.
right now we're really coming up with new ways to leverage our collective expertise in chromatin biology, drug discovery for transcription factors, and mass spectrometry proteomics. So this is kind of three areas where I think there's plenty of scientists out there who might have one of these areas of expertise, but having a team that really brings together these very different ways of thinking about drug discovery has made us really unique. So our big drive right now is hit to lead, working on our own internal drug development pipeline.
Cool. And hit to lead, have you already started working on animals, or that's like a
No,
next?
that's
Okay.
definitely our next step. Yeah, that's kind of the dream with kind of what we're doing now is that we will take a lead candidate, a lead compound through kind of ADME and all the traditional med chem steps, and then get it into animal models.
Pretty cool. Pretty cool. And one thing I'm curious about is, so you have the pretty unique approach how you aim to cure diseases or improve the lives of humans. But what is the competitive landscape? Like how it is so unique than the traditional way of doing research?
There are some companies that are also very interested in, drugging the undruggable.
Right,
There's definitely some other therapeutics companies that are focused in this space. A lot of them are just using kind of general approaches. A lot of those companies are just, kind of the Protect style companies.
right,
Protects, protein degraders are extremely hot right now. So a lot of those companies don't really necessarily know anything specific about transcription factors.
right,
So that gives us a little bit of an edge. And then some of these other companies, there's a company who purportedly knows another piece of the structure puzzle in some transcription factors. So I had mentioned how transcription factors are like largely intrinsically disordered except for the DNA binding domain. There's some other companies who say that they have found another domain that could be a potential druggable pocket.
right.
It's not present in all transcription factors. So that kind of gives us an edge too. We're agnostic to the amino acid sequence of the transcription factor with our platform, but it's just such a wide open space that I really think there's room for therapeutics from multiple approaches. There's almost 2,000 something known transcription factors, so quite a few.
Wow.
About 1,000 transcription factors. Over 400 of those we know are oncogenic. They cause cancer. Of those over 400, that we know the identity of and we know that they cause cancer, only 18 have an FDA approved drug.
Wow.
So
Okay.
there's a very large target space here for multiple companies.
Is there any limitation of this approach? Where even Moderna has very agnostic platform approach, but mRNA, it still has its own limitations. So with your approach, is there any limitation that you cannot touch this therapeutic area or you cannot focus on this type of disease?
Yeah, I don't think we have limitations as far as what types of therapeutics. So right now we're kind of mostly focused on small molecule therapeutics,
Right,
but there's no reason the platform is agnostic. We could be looking at cell therapies like CAR T cell approaches. We could also use it strictly for phenotyping to
right,
find biomarkers or candidates. We're not, as a platform company, we're not so much unlimited in the sense of like Moderna as far as, just swap in and out what DNA sequence you want. That's a pretty awesome inspiration, I think, for all of biotech. I think the
100%. Yeah.
success of Moderna and, the scientific community in the face of COVID, I think has inspired a lot of investors to reconsider the biotech space, the biomed space. So
Pretty cool. Speaking of Moderna, they are a digital biotech. They spend millions of dollars creating custom digital workflows. And I'm personally very passionate about the data companies generate. How can you help them digitize? How can you help them make research more reproducible? How can you automate their workflows? So, what are your thoughts about the whole leveraging your data to do better studies and the big operate like a data science company, not just as a wet lab company, right? Because ultimate goal, even when you run experiment, is to generate quality data. To
yep.
so, what are your thoughts about the importance of digitization, automation, and all the lab-based companies?
Oh yeah, digitizing, automating, storing metadata, and storing even the data itself in a way that is useful, I think is all incredibly important. Not just for us as kind of a blended company, a blended wet lab, dry lab company, but I think just in general, it's really crucial to have all the details of your samples, how they were manipulated, where they came from, their origin, having all that metadata captured in a place that can be easily accessed by anybody, not just your wet lab team who is generating the samples and doing the physical manipulation, but also your dry lab team to have access to that metadata so that when they're analyzing, they can look for batch effects or they can
Right.
quickly pull data based on these metadata characteristics. They could pull all of the data from, some specific cell line, or they could pull all of the data across one of the compounds. So I think all that is hugely important. I actually think there's a huge space for improvement in a lot of these things, as you know, because there's just not a lot of good tools out there right now, currently, that can help you do these things. I think a lot of companies are homebrewing their own internal solutions to this, which just seems dumb. It's like everybody trying to reinvent the wheel for their own internal purposes. So I think that's all really important. And I think some of like the big server companies, Amazon, Microsoft, Google, like they've all kind of realized that this is a huge area, because similar to all these companies, all these labs homebrewing their own software for sample managers or electronic lab notebooks, LIMS systems. Also, all of these labs and companies are homebrewing their own servers to do their data analysis and host their data. So I think, AWS pipelines are getting really popular, that you don't have to have your own cluster and maintain your own cluster on site. You can just spin up whatever instance you need on AWS for your data analysis. So I know that those are kind of tools that have helped us as a company move a lot faster using those kind of services, because we don't have to worry about, in this great cryptocurrency world we live in now, we have to try to get GPUs and CPUs and put together a server. We can just spin up instances when we need them.
Right. Server and also the tools. I see many of the companies still their data is siloed, because they don't want to spend millions of dollars for, it's reasonable. You don't want to spend millions of dollars, hire developers, create custom solution. So I think there's quite a bit potential to make all of these companies data science companies. And I think that will move the needle quite a bit.
I totally agree. Yeah.
Cool. So Lindsay, it was really fun talking to you. I learned quite a bit about your company, Proteomics Space, and all the great work you are doing. Really appreciate it.
Yeah, thanks so much for having me, Guru. It was really great to kind of tell a little bit more of the downs than just the ups of starting a company. And yeah, I hope some scientists out there really find some inspiration and realize you can do it too. Yeah.
Do you have any message for women in bio? Because potentially it's a multi-trillion-dollar industry. We need equal representation.
We absolutely do. And not just women, all kinds of underrepresented minorities, because we need different ways of thinking about things. And I think diversity of ideas is really increased by just having diversity in the workforce, diversity in the entrepreneur space. So I would say, if anybody out there is from one of those underrepresented groups, like there are resources for you too, specifically for you, resources on how to start a company, resources for investors and funding that are specifically interested in helping you as a founder create a company and get in this space. So
totally take advantage of every unfair advantage you have, right?
Yep.
Hey, Lindsay, pleasure talking to you as always, and talk to you soon.
Yeah, thanks so much. Have a good one.
Bye.